handing this to each other. So, oopsie. There. Okay, so thanks everyone for being here. And today we're gonna be giving you a little bit of a story that goes back to like 2013, actually, about Duxbury-Kingston-Plymouth, which we call DKP. So we're like pet acronym for debt embayment. So the story is about an investigation that we've been wanting to run on eelgrass loss. Most of you know, Duxbury-Kingston-Plymouth is an embayment made up of these 3 bays, and it's relatively not very populated. I mean, it's would call it like it's semi-urban in my opinion, but it is the Bay of Duxbury there to the north has barrier beaches surrounding it, and where it says Clark's Island and Squamish Neck that belongs to Plymouth. So there are some town boundaries there. But in 2013, we had reports of eelgrass leaves floating around by some fishermen who also were noticing that there were patches of eelgrass where it used to be that they are not observing eelgrass anymore. Eelgrass had been surveyed by MassDEP in DKP starting in 1995, and we were observing lots of losses over time. So we decided to launch an investigation into what is happening. By we, I mean MassBays and Division of Marine Fisheries. And from then on, the reports went to DMF first, and DMF started with the investigation around that time. And then it kind of grew bigger. It involved North and South Rivers Association plus volunteers. So over time, it grew into a bigger effort. It's just a little bit about DKP. It has a high tide of semi-diurnal tide, of 3.2 meters. And the opening there is about 1 mile wide, and it has a quite good natural flushing. There's plenty of riverine discharge. There's Eel River and Town River in Plymouth. There's Beck River and Jones River in Duxbury and Kingston. There's also lots of activity, both recreational and commercial, including boating, aquaculture, oyster aquaculture in Duxbury in particular, fishing, and also in terms of uses, lots of boat moorings. And there's also wastewater treatment discharge as well from the wastewater treatment plant in Plymouth. So there's a host of things that are, running around it, surrounding this embayment. And so there was a lot to dig into in terms of trying to figure out what the causes are of this eelgrass loss that we were noticing. So now I hand it over to Jill. So I'm going to stop sharing and.
Nope, you're good. Keep it shared. And then.
Okay.
And I'm just going to request remote control and you can hit OK.
Oh, okay, great. All right.
There we go.
Okay.
Okay, great. All right, there we go. Yeah, thank you for that introduction. I know several people on the line have seen and heard presentations about what's happening here over the years that we've been interested in this area, but it's still very important and there's still a big question mark over certain parts of it. So that's why we're bringing it back to this audience now. So after receiving those anecdotal reports of losses by fishermen back in 2013, we first looked to readily available data about eelgrass, looking at MassDEP's eelgrass maps, just to see if they aligned with those anecdotal accounts. And they most certainly did. And as you all know, DEP's mapping program goes back to 1995, and it's updated roughly every 5 years. Here are 3 of the time steps for DKP. On the left is 1995, which is showing an estimated 2,200 acres in green. And then on the right, by 2017, there were about 945 acres, so a 57% loss in that time. There's an unofficial eelgrass map out there. It's not shown here. That was generated using black and white imagery from MassDOT in the 1950s. And that had an estimate closer to 4,000 acres at that time. So that puts us closer to a 75% loss since mid-century. So these are very substantial losses that we're talking about here. And with DEP's mapping program on a 5-year cycle and their aerial image methods providing only a moderate level of detail, it became very clear that there was a need for more detailed and more frequent mapping of eelgrass, along with an investigation of stressors. So that's really what brought us to the investigation I'm going to talk about for the next few minutes. I was working at MassDMF at the time, and that's when we partnered with MassBays, and that's when Preston and I really started working together on eelgrass stuff. So that's what I'll be talking about more here. Let's see. Okay, so for more detailed mapping, DMF used 2 different acoustic methods. We used single beam echo sounder, which pings sound directly underneath the boat, and it measures things like, well, it could detect features, and it can also measure depth and bathymetry directly under the boat. And also multi-beam side scan sonar, which also uses sound, but it pings it sort of in an array of angles out to the sides of the boat. So you get sort of a swath of imagery underneath the boat. And what you're seeing in the picture there on the right is an example of side scan sonar results. Each of those gray swaths is about 200 feet across in reality in the field, or 100 feet to either side of the boat. And that fluffy sort of cloud-like texture that you can see there, slightly lighter in color in the grayscale, that is a classic eelgrass signature. So that mapping was conducted in 2014, 2016, and 2017. The 2014 mapping was the most intensive of those 3 years and covered most of the embayment, and that resulted in the eelgrass layer that you see on this map in green. Then in 2016 and 2017, we did more of a targeted approach where we did these long swaths that were designed to cross several beds. And this was more of like a rapid method of determining general presence and absence across those beds. And those gray swaths are shown here in this map. And then the results of all 3 mapping efforts are shown here, where the green is still the 2014 result. And then the blue portions indicate areas that disappeared in 2016. And then the red areas disappeared in 2017. There's a pink area in Upper Duxbury that was inconclusive. The data didn't look great. The imagery was somewhat poor, but it was expected to be gone. And then unfortunately, as we now know from DEP's more recent mapping, that's most likely the case, that it did disappear. So we're talking about a pretty rapid change happening in just a few years, some major portions of the beds disappearing. Then we wanted to get a sense for the rate of loss between each mapping interval to sort of predict when total loss might be realized. And we used loss rates over the entire time series, how many acres were lost between each mapping step. And if we used the most recent rate of loss on the bottom row here, that was the loss that occurred from 2012 to 2014. That was about 400 acres per year lost. We would have total loss across DKP as early as 2016. Fortunately, that did not come true. If we look to the average loss rate across all of the mapping years, that loss rate was 132 acres a year, and that would lead to total loss by 2021. Fortunately, that doesn't appear to be entirely true either, and the median loss rate was closer to 27 acres per year. And under that scenario, all eelgrass would be lost by 2051. So sort of a depressing picture here, although we are hopeful that the first 2 scenarios didn't play out. Oops, oh boy, there we go. So then to better understand the nature of the loss, in 2016, we reinterpreted DEP's raw aerial imagery, this time to differentiate dense grass from sparse grass. So that's not something DEP does. They draw their boundary around the entire meadow, including anything that they can detect visibly. So this time we wanted to see if there was any sort of pattern in terms of density. For example, for this image, this is a Nahant image, we would have parsed out these areas where you can clearly see it's less dense eelgrass. These would have been parsed out separately from the denser meadows. And the sort of the difference between the 2 classes was dense grasses where we had greater than 50% aerial coverage when looking at the imagery at a scale of 1 to 1,000, and sparse areas were less than 50% coverage. So this was done for all DEP imagery from 1995, 2006, and 2012. And the results are shown here. And I'll just give you a second to orient your eye here. So that the upper left is 1995, dark green is the dense grass, light green is sparse grass. Upper right is 2006, lower left is 2012, and lower right is 2014, but that's just DMF's sonar data, that's not DEP's data. So what we're seeing here is dense grass declining embayment wide over the time series. Between 1995 and 2012, 43% of the dense grass was lost. And then in the bottom right, that 2014 map showing the sonar results, you can clearly see, between 2012 and 2014, it looks as though those more sparse beds have just completely disappeared. So the takeaway here is that the loss in this embayment really does seem to follow a pattern of the meadow transitioning from dense to sparse to gone. We see that over and over again, where it sort of follows that pattern. Oops, all right. And the final component of this early study was exploration and analysis of stressor data. And that included a search for data on abiotic variables like water quality, physical impacts, sediment burial, and then also biotic variables like wasting disease, epiphyte cover, invasives like green crabs, and predation. And I'll note, we didn't go deep into nutrients at this time because there was a lot of work being done on that topic. There was a nitrogen load study being completed at the same time by UMass and DEP, so we sort of didn't touch nutrients at that time and focused on everything else. We were sort of hoping to look beyond water quality to some of the stressors that are less often in the spotlight when we're talking about eelgrass loss. And the general take home from this effort was that we did not find a single smoking gun, but several concerning variables. So in terms of water quality, sea surface temperatures, chlorophyll A, and stream flow were 3 variables found to have significant increases over their time series. These were Center for Coastal Studies data. And again, our analysis stopped at 2014. It was from data from 2006 to 2014. So that we could certainly stand to do this again and update our analysis. And Amy, who I know is on the line, might be able to speak to this too, if she's had a look at the data since then. There was also evidence of impacts from dredging, aquaculture, mooring fields, and also propeller scour in some of these very shallow shoaly areas. And those we do think explain some localized losses, but we really don't think those physical impacts are responsible for embayment-wide losses, especially on that that pattern that I was just describing is not really a classic physical impact, a physical impact pattern. We do think that sediment dynamics might be very important here. There were no data sets available to us to help us investigate it, but we have heard many accounts of areas that have shoaled over time. Fishermen that have been out there for decades have talked about meadows that have buried over the decades, other areas that have completely scoured. And some people have talked about areas of fundamental ecosystem change, such as an area that went from a sandy bottom to all of a sudden being a dense, hard mussel bed, or vice versa. So it's an extremely dynamic embayment and very interesting in that way. And there may be many natural influences that are happening and that are driving sediment dynamics and movement. There's a lot more on all of these data sets, a thorough analysis in our report, which I believe is linked, I just can't see it in my view, linked at the bottom of the page. Yeah. So all of that said, and then at the bottom there, we tried to dig into data sets on a handful of other really important variables, but there were just no data. So at the time, no one was looking at wasting disease there, or epiphyte cover, or green crabs, or there was some very limited green crab work. It's very hard to track down data on predation from like waterfowl, and the local stakeholders felt that herbicides could be something of importance too here with local golf clubs and golf courses and things of that nature. But again, it's just really hard to track down the data to go with that. And we did look into ice scour a bit. There are lots of accounts of large ice flows kind of moving around certain years, but it's really, there's not a time series that we could actually look at for that either. So all of that said, this early effort that we made, DMF and MassBays, really brought to light the need for more data, more frequent data on eelgrass presence and condition, which ultimately led to this formation of citizen science methodology that Sarah's going to get into next. But then also more need for targeted water quality and sediment data. And excuse me, Preston is going to talk about that a little bit at the end too, some work that we're doing right now and hoping to continue into the future. So hopefully that sets the stage for what we'll hear for more current efforts that Sarah and her team at North and South Rivers are doing, and Forrest with analysis of the data. So with that, I'll hand it over to Sarah.
Okay, I just requested. All right, here we go. So now I have control.
Yeah.
So as Jill mentioned, we had data from the aerial photography, but it was 5 years in between those flights. And that wasn't quite enough detailed information year after year to know, did all of this eelgrass disappear slowly over those 5 years, or has it disappeared in one acute event? So we needed a mid-level between that and something much more detailed like seagrass net, the tier 2 level monitoring. So we decided to create a citizen science protocol, and we got a lot of inspiration from the work that Hillary Nichols had been doing with her hexagons. So we pulled together a bunch of experts. Many of you here were in that audience. And we also brought in a lot of the local people who had been concerned about the eelgrass to help us create a protocol that would work well for citizen scientists, but would also gather the information that we would need. We went through a lot of different trials in terms of what we would use when we were out in the field, and finally developed an eelgrass blitz kit that could be brought out on a smallish motorboat. We typically say 16 to 25 feet, like Boston whaler or a skiff. And the kit contains a camera, an onboard camera that's connected to a quadrat platform that's sort of a prism or a pyramid. And that platform can be tossed overboard and take photographs of the bottom. And then we also have a kit for analyzing some of the eelgrass biology a little bit closer. All right, so let's see. I'm hitting the arrow, so it should go, right? Is it advancing at all?
No.
Try a mouse click instead.
Yes.
A mouse click, okay.
Mm-hmm.
Hang on.
Yeah.
There we go, perfect. All right, so first we needed to just determine where we were going to be collecting our samples. So here's the bay, and the bay was divided into Plymouth and Duxbury, and then using those, and those are the 305B sub-embayments, and then based on the DEP polygons, we came up, Jill really did a lot of this work, came up with places where eelgrass had been identified in 4 to 5 of those years, or in 1 to 3 of those years. And it was bounded by minimum and maximum depths for historic eelgrass. So sites that were between 1.5 meters to 5 meters. And then there were also sites that were removed that were inaccessible. After that, we also added in, sorry, I can't see the bottom of it, places that eelgrass had also never been identified, but would be candidate sites for eelgrass based on the conditions there. Oop. So we ended up with about 250 sites randomly distributed throughout those areas, and that was reduced down to 120 in the subsequent years, just in terms of trying to have manageable effort. So we would have training on land beforehand. You can see there 3 of the kits. And of those sites, 119 are indicator sites. So is that right? Yeah. So that means that in addition to dropping the drop camera over the side and taking a photograph of the bottom on all 4 corners of the boat, we also would collect a sample of 3 eelgrass blades per corner and measure their length and their width and assess them for epiphytes and for wasting disease. And in addition, all of the metadata is also collected at each site. So we do secchi depth and note the wave condition and the wind. And the eelgrass cover based on the screen shot is binned in real time. So we look at the screen and try to determine whether the percent coverage is 0, 1 to 10, 11 to 30, 30 to 75, or 75 to 100. And then that data gets reanalyzed. Those photos are looked at on a much nicer screen on someone's laptop later to do QA/QC on the assessment of those photographs. And every year we've had an eelgrass intern who has helped run the project with us. This year, Taylor is going to be doing that work. And so at those indicator stations that I mentioned, we also collect samples for leaf area index, and then the epiphytes and the wasting disease. You can see someone in the lower corner measuring that out on the side of the boat. And now, as of last year, we have been logging the data in a web app, iSeagrass, instead of writing it on paper data sheets, which has been immensely helpful because entering all the data is very, very time consuming. And this way it's already entered and can just be QA/QC'd. We try to resample the same locations every year. The protocol for getting to those locations involves using the GPS coordinates to navigate to the site, throwing a marker buoy and then anchoring down on the site. And we are always trying to get within a 10-meter radius of the true GPS location. Some volunteer boats are better than others. We do use citizen science boat captains as well as citizen science volunteers. So we try to recruit boats from the local area, people who already have the right kind of boat, who are around Duxbury, Kingston, and Plymouth, and are willing to spend a day or two with us. So it turns into a multi-day eelgrass blitz where we have a few boats going out, hopefully each day, with a chief scientist aboard, as well as a couple of volunteers to conduct the work. And here are some shots of what we get back from our drop camera. So you can see we've got some sparse eelgrass, we've got some dense eelgrass, we've got some algae that has the same sort of long, thin signature, that that cord algae. We've got some shell hash, and then of course sometimes we see crabs. So it's just an example of what these look like. And you can see that we have a slate that's attached to the quadrat that tells us what site we're at. And then we came up with making hash marks on the slate to know which corner of the boat we're talking about. And we discovered that it's easier to erase hash marks than add them to a wet slate. So we actually usually do it backwards. We start with 4 and then we work our way down to 1. So there's a little bit about iSeagrass, the app. It was developed with a lot of effort from Forest, and it matches what we would be writing on the data sheets, collecting all of the information about the boat and where we are and the secchi depth. It's pretty fail-safe, I would say, and really the only concern is making sure that everybody has charged up phones. So we try to have charger blocks with us when we go out in the field. And it's great because it's open access. And the other great thing about it is that we don't have to wait to look at a preliminary map of presence and absence. So this is just an example of the data that it can generate, where you're seeing that there were 179 stations sampled, and you can see where we found eelgrass, where we did not find eelgrass. Think back to what Jill was showing. It does match those patterns that we're seeing in the other mapping efforts. So in terms of actually doing this, the protocol is pretty easy to implement. We haven't had any issues with anybody not understanding what is supposed to happen. Originally, we were collecting shoots using an anchor, throwing a small anchor overboard and kind of pulling it in. But the problem with that was that we would do throw after throw and not get anything, and then occasionally pull up an entire big patch of eelgrass and feel terrible. So now we are using snorkelers because none of the sites that we're working at are particularly deep. It's pretty easy to free dive down and pick up a couple of pieces of grass. It's definitely crucial to make sure that everybody is trained and knows what they're doing, that there's somebody on the boat who is well versed in the protocol. Our biggest issue is definitely getting enough boats. We can usually get enough volunteers, but the boats are really where we end up being limited, and that limits how quickly we can do the protocol. And in some cases, we actually have to, last year, we were not able to complete the full list of sites. We do sometimes also have weather delays, of course, and there are places in the bays that are very shallow. So when we're deciding where to go each day, that is something that we really have to factor in, because we want to work as much of the tide as we can, but also not get stranded on these shoals and sandbars that are out in the bay. And she'll actually see me hauling the boat across a sandbar in the lower right-hand corner. So, but it does generate a lot of data, which is great. And Forrest is going to tell you a little bit about that data next.
Hi, everybody. Right, so I just requested control.
Yeah, you have control.
Okay.
Okay.
Awesome. Yep. Clicking and advancing slides. Thanks all for inviting me to the STAC meeting. I'm excited to share this data that Sarah just described, how we collected it. Yeah, it's not quite hot off the presses, but it's exciting to have this group take a look at it. And yeah, I'm really looking forward to the feedback. So as kind of this presentation is set up, this citizen science project and the data collected with it was really informed and kind of the next step from the analyses that Jill and Catherine did in 2016. And so the kind of questions are very similar. We're still interested in trying to understand better the nature and the causes of changes in eelgrass distribution that we've been observing at DKP. So the first section of these results, I'm going to talk kind of about from 2018 to 21, where is the eelgrass persisting and where is it being lost based on our drop camera data from the station. So kind of spatial trends in eelgrass over these last 4 years. And then the second part of the results section, I'm going to look at some of the predictors of eelgrass decline that we were measuring. Sarah mentioned epiphyte and mason disease coverage, and also sediment and turbidity at our stations, and trying to look at correlations between those and then areas where we've lost eelgrass over the sampling period. All right. So start with kind of the spatial analysis. Here's DKP embayment, and in green here, I've just showing the eelgrass polygons from kind of the 2012 through 2016 mapping. So that was really the most recent aerial mapping that was done prior to the start of the citizen science survey. And sorry, the green areas are where eelgrass was mapped in those years. And then you can see these points here. These are the sampling stations that were visited by the volunteers over the 2018 to 2021 time period. And each point represents a single station that was visited at least once, but usually multiple times in these 4 years. Most of the stations were visited every single year. I'm just going to go into so I've color coded our stations. And the first color I want to talk about is these green stations. And I've circled kind of clusters of these here with a green outline. And these are stations where we observed eelgrass every single time we visited them between 2018 and 2021. So I'm kind of considering these are stations where eelgrass is persisting. You see these areas of kind of clusters are around Parks Island and between Parks Island and St. Christian Neck. And there's a little station here and around the inner kind of inside of Plymouth Beach that's also been persistent. Okay, the next color of stations are stations where we haven't observed eelgrass in any of the sampling times we've been out there. And so these are areas of where we're pulling eelgrass absence. And so these are still, I think nearly all of these areas were mapped at least in 2012, and most of them were mapped as recently as 2016. But these are areas that were apparently lost or eelgrass disappeared before we started sampling in 2018 and have not recovered since. And you can see kind of up in Duxbury Bay and kind of the inner part of Parks Island, we see these areas where we haven't seen any eelgrass in the last 4 years during our survey. And then finally, there's this kind of majority of the eelgrass meadows in DKP where we're seeing a real mix of stations. So both absence and presence, but also want to highlight, it's a little hard to see, but there's these yellow colors, and these are stations where we observed eelgrass in some of the years, but not all of the years we visited stations. And so these are areas of, we're actually seeing kind of dynamic change in eelgrass during our sampling, point between 2018 and 2021. And so now I'm just going to go in a little deeper in exactly what these changes were. Are these losses or gains? And so in this next slide here, I've kept kind of our orange circles around these areas of kind of change. But now I'm just focusing on those stations where we saw either a gain or a loss, and I'm coding them by whether they were gains or losses. So a green dot means that eelgrass was absent in the prior year, and then the next year we went back and we saw eelgrass that year. So that would be a gain. and then the opposite obviously would be a loss. And since this is over 4 years, you can note that some of these stations actually have seen both gains and losses. There's a little orange X for loss, which is either above or below the green circle of gain. So sometimes we see eelgrass almost blinking in and out of these different stations. And so I went a little further and kind of started color coding these areas based on whether we're seeing mostly losses, and these are the kind of dark purple circles now, or whether we're seeing gains, and the kind of 2 green circles here are places where we've just seen eelgrass gain and not losses. And then I've kept the orange circles around areas where it really seems to be a mix of gain and loss. And there's no real clear trend. And just, a couple, the spatial patterns are, this isn't necessarily a formal analysis, but one of the spatial patterns is that most of these areas we're seeing a lot of these losses over the last 4 years do tend to be kind of in the interior of the bayment. And then kind of in this, towards the mouth at least, we see gains, or at least areas where they're kind of combination of gains and losses. So it seems like maybe there's going to be there is some kind of spatial trend in the changes in eelgrass, but definitely more formal analyses I think are needed, and I definitely love feedback on how to ask those or what methods to use to answer those kind of questions. And I just wanted to, also just mention quickly in the bottom left, over the 4 years, we observed 20 instances of eelgrass loss at our stations and 14 instances of eelgrass gain. And they weren't, I'm not showing this data, but they weren't necessarily clustered in 1 year. There wasn't like one great year where eelgrass made great gains. They were kind of distributed pretty evenly. And that's the same for the losses. Typically we saw between 7 and 13 stations where there was loss in any given year. So they were distributed fairly evenly. Okay, so kind of to summarize, again, the spatial trends we've been seeing in the last 4 years, I think one of the take homes is that there really has been widespread change in eelgrass that's continuing throughout the embayment. So that change that was first observed in 2012 through 2016 does seem to be continuing, and this is including new areas of eelgrass loss since those analyses. And then I want to highlight again that these losses do seem to be, for the most part, concentrated in the interior of the bayment. And then finally, the stations where we're monitoring are areas where they're kind of considered core eelgrass habitat. They're areas where we observed eelgrass, usually most recently in 2016, and they're really consistently observed from 1995 through 2016. So it is very concerning that we're not seeing eelgrass in these areas, and also we're not seeing any recovery in these areas. And I just wanted to stress that point, and especially these areas where we're not seeing recovery again tend to be in the interior of the bayment. Okay. Well, great. So the next part of the next section of the results I want to go into is looking at how those predictors of eelgrass declines that Sarah was mentioning, the sediment, the Secchi disk to measure kind of the turbidity of the water, and also some of the biological variables like wasting disease and epiphytes, and the condition of eelgrass itself, whether that's kind of the size of the leaves, leaf area index, or the kind of percent cover, whether that is giving us any hint or kind of predicting or preceding some of the changes we've been seeing over the last 4 years. So how I decided to look at this is I just looked at stations where either the eelgrass persisted from 1 year to the next, or where it was lost from 1 year to the following year. And I coded those stations as either a persistent or a loss. And then I looked at the data from that first year. So the year prior to the loss or the year prior to the persistence. and compared how was comparing our different predictor variables between stations that either persisted or were lost. And again, we kind of I've split our kind of environmental or biological stressors into these three categories. One is the Eelgrass condition, which is percent cover and morphology. And then we have our biological stressors, which are epiphytes and wasting disease. Then finally, environmental conditions are turbidity and sediment type. So I'm going to run through kind of how these different variables compare between stations where we saw eelgrass loss versus eelgrass persistence in the following slides. All right, so the first predictor I'm going to look at, or stressor, or really this is kind of a predictor looking kind of a signal, whether we're going to see a signal between eelgrass percent cover the year preceding and whether we saw eelgrass the following year. So this kind of, this question was really inspired by one of the findings of Jill's and DMS report in 2016 that we saw eelgrass going from kind of dense to sparse to then gone. And we wanted to see whether we saw that a similar trend in our data from 2018 to 2021. And particularly what this would look like would possibly be maybe stations that have lower percent covers the preceding year than the following year having loss. So on this figure here, on the x-axis, looking at our eelgrass percent cover bins, Sarah kind of described earlier, and then on the y-axis, looking at the proportion of observations in each of these percent cover bins, in which eelgrass was either lost or persisted at the stations when they were sampled that following year. So the key here is I'm kind of drawn in a little red line to split the graph. I think there's kind of a separation here, and that's really, you can see that nearly half of the nearly half of the observations of either 0 or kind of this really low percent cover category were at stations where eelgrass was then lost the following year. So this is these orange bars. They make up a much greater proportion of those observations than when we have higher eelgrass percent cover bins. And you can see now that the losses of the orange bars are a much smaller proportion. Really only about 3/4 of the observations are where eelgrass was lost. So this is kind of suggesting that there's maybe that same signal of kind of low density or patchiness then is signaling losses that are coming. Another way to kind of look at this data is to use an NMDS plot. So here the green and orange dots, these are different stations which either were lost or persisted. And I'm kind of plotting their eelgrass percent cover bins in two-dimensional space. So the black points here of different shapes, these are where the different bin categories of cover fall within that space. And we can see that the kind of center of the point cloud where of eelgrass loss stations, that tends to be closer to the lower percent cover area in this figure. And in comparison, the center of the point cloud of persistent stations, that tends to be towards the higher percent cover stations. So again, while there's some overlap in the point clouds for sure, it does suggest that again, this visualization is again suggesting that these lower percent cover areas are maybe a signal of upcoming losses. All right. So the next category is still in the eelgrass condition, but now I'm looking at morphology, and this is reef area. So now on the x-axis here, showing whether that observation was associated with a station where eelgrass was lost or whether it persisted the following year. And the y-axis here is the area of the leaves in centimeters squared. And really there doesn't appear to be a strong trend here in terms of the the size of leaves and whether that area was eelgrass either lost at that station or whether it persisted. So it doesn't seem like the eelgrass leaf areas maybe is really signaling changes very strongly in the embayment. So now moving to the biological stressors, looking at epiphyte cover now, and epiphytes were ranked on kind of a scale, a four bin scale from none to low to medium high, high being the most epiphytes, low being still present, but the least amount, and then none being the absence of epiphytes. And on the x-axis of this figure, we have the epiphyte rank, the different bins. And then on the y-axis, again, this is the proportion of observations in each of these epiphyte rank bins in which eelgrass was lost or persisted the following year. So losses are in orange and persistence is in green. And what I want to highlight here, again, I've kind of put in a red line to kind of highlight that the split. Well, I think maybe there's a split in the data, and we're seeing that there's very few observations where epiphytes were categorized as none. So the absence of epiphytes were from stations where eelgrass was lost that following year. And in comparison, when epiphytes are present, we do see that there's more of those observations are from stations that are lost the following year. So there's seems to be this split between the absence versus presence, and this makes some sense because we know epiphytes can be a stressor, but it's also interesting that we're not seeing a really strong trend when epiphytes are present, where we're seeing a greater proportion of observations, for instance, in the high epiphyte rank, where the eelgrass is lost from that station the following year. And that's kind of what you would expect, I think, if epiphytes were a really important stressor. You'd expect to see these orange parts of the bar chart expanding as we're going from low to high, and we're not seeing that. So that makes me think that perhaps epiphytes aren't, well, they may be playing some role, aren't necessarily a key factor behind some of the declines you're seeing. And just to add to this point, now again showing on this figure the epiphyte ranks on the x-axis again, but now we're just looking at, rather than the proportion of observations, the frequency of observations. And my take on here is that most of our observations, both from eelgrass stations where eelgrass was lost and from when they're persisted, the majority are in the low to medium epiphyte bin. And again, this is, I would expect if epiphytes were a major problem in the embayment, we could see more of observations in the high epiphyte bin. And now moving from epiphytes into wasting disease, just like similar to epiphytes, wasting disease was binned into these 4 bins of absence, low, medium, and high. And again, high being the most wasting disease on the leaves, low being the least, and then none is the absence of disease on those plants. And again, looking at the proportion of observations on the y-axis, and persistent stations where eelgrass persisted in green and where it was lost in orange. And I really not seeing a strong trend in any direction for this data. So this makes me think that there really isn't, wasting disease, at least in this embayment, isn't playing a strong role in the changes in eelgrass survival, at least from 2018 to 2021 when we were sampling. And just to add to this point, I want to show this figure, and now similar to the epiphytes, again, the wasting disease bins are on the x-axis, but now I'm looking at the frequency of observations on the y-axis. You can see that most of the observations of wasting disease were in the low category, and really the vast majority were. And they're also distributed, as you can see in proportion, really evenly among the survival, whether eelgrass survived or not. So these two kind of together, the fact that most wasting disease was low and there wasn't really a strong trend in the proportion of stations that survived in these different bins, suggests that wasting disease is not an important player, at least in terms of the changing eelgrass distribution in Duxbury in DKP over the last 4 years. All right. So now moving into our looking at our environmental signals. And the first one I'm going to show here is turbidity, which I've plotted as Secchi depth. So on the x-axis here, we have whether the following year eelgrass either persisted or whether it was lost. So the green persisted or lost orange, and then the Secchi depth from the preceding year at these stations is on the y-axis. Just a quick review on Secchi depth, the larger the depth, so the farther down in the water column, you can see the Secchi depth, the Secchi disk, the less turbid or more clear the water is. So as Secchi depth is increasing, turbidity is decreasing. And what we do see here is there does seem to be at least a slight trend. I haven't run formal statistics on these data yet, but it does look like there's maybe a trend where stations where we saw losses the following year, the Secchi depth tended to be lower, and so the water there is more turbid than at stations where eelgrass persisted. And this makes sense because we know eelgrass requires light to grow, and so the more turbid the water, the less light is able to filter through the water column and reach the plants on the bottom. And this can be an important stressor. So this is suggesting that water quality, similar to Jill's report, it's kind of is in agreement that water quality may be an important factor in DKP in terms of the changing eelgrass distribution. And then finally, the final signal I want to talk about is sediment type. So on the x-axis here, I have our different sediment categories. And there are actually only 3 in stations that persisted and were lost in DKP. We only recorded 3 different types of sediment, which are either mud, a combination of mud and sand. And then again, the y-axis here is these are the, I'll make sure I say this right. It's the proportion of observations in each of these sediment categories in which eelgrass was either lost or persisted the following year. So lost is in orange, or green is persistence. And I think what's interesting here is we're seeing a trend where we go from mud towards sand of increasing persistence. Or so when you think about what mud versus sand sediments mean for eelgrass, often mud sediments are finer sediments and they tend to be more anoxic. And we know anoxia can be a stressor for eelgrass, especially when combined with water decreasing light availability or increasing temperature. So these kind of stressors can work in tandem to, in other systems they've been demonstrated to work in tandem to cause eelgrass loss. So it does make sense that we're seeing more stations lost where the sediments were mud versus where sediments were sand and maybe are more, the plants aren't so oxygen stressed, which is promoting their persistence. And also want to show I want to show the frequency of observations where we're seeing mud versus sand, just to make the point that many of these stations where we've been seeing eelgrass, since 1995 through 2012, about a third of them were in mud sediments. So it's not that just that only a few stations are in mud, and it's kind of a really minor sediment type. It really is a fairly common sediment where eelgrass has at least historically been growing in DKP. So it could, again, the fact this suggests that it could be the sediment type could be contributing to some of the declines that we're seeing in the embayment. And so just to summarize our results, in terms of eelgrass percent cover, the results from our citizen science data are supporting or in alignment with what the aerial mapping found in previous years, and that we're seeing this pattern again of eelgrass growing maybe from dense to sparse to gone. Evidence that that pattern has continued. Oh. Yes. And then the second portion I want to highlight is that there do seem to be some environmental stressors like water quality and sediment type that are showing up as possibly correlated with these eelgrass losses in the embayment, which again suggesting that maybe these are some important factors to continue to focus on, or these are some stressors that are at least we can't definitely say definitive, but they seem to be associated with the declines in DKP from 2018 to 2021. And with that, I will pass. I think we're gonna, transition to the questions for discussion. So Preston, I'll give control back to you.
Okay. All right. I think I have control now. So thank you, everyone. Dan, do you want to kind of move this along or take this over? So this is like a set of questions for discussion. Then we have a couple of slides with next steps at the end. So, but I think it's a good place to stop for this.
Sure. Yeah. I mean, why don't you go ahead, Prasad? You seem to be in touch with the questions and able to facilitate. I don't mind that, unless you prefer that I would facilitate.
No, it's we can tag them anyway. So basically, these are some of the questions that have been coming up to our mind based on these findings. And thank you, Jill, Sarah, and Forrest for this really great summary and actually detailed summary of what we have been finding. And I also wanted to add that this whole project, since it started, was funded both by EPA through our MassBays project. We had some supplemental funding, but as well also mitigation funding that was provided via DMF. So it's great that we had those opportunities, and hopefully we'll be able to get some funding to continue the funding so that we can continue this work in the next few years. But so basically we wanted to get your thoughts on the methods and data acquired today from the Rapid Assessment Survey, that's the Citizen Science Survey. For example, should the assessment be continued annually? Are there any changes we should make from the volunteers, from that volunteer survey? Based on the findings so far, where should our effort or investment be focused? Is there value in eelgrass transplanting, for example? What methods can be applied? And what are other data and information that we should gather to enhance the investigation? So we could go kind of question by question, but we could also, kind of keep these in mind. Basically, we want to get your thoughts on what our results are showing so far. Are we headed in the right direction? And what would be the next steps to kind of start honing in now onto the actual causes of these changes? So.
I'd love to just jump in and kick off the conversation a little bit with something that I've been thinking about in watching these presentations, which have been really, really interesting. I'm really curious about whether the long-term changes could be mostly due to movement of sediment. And the reason I'm curious is that I've been looking at wind observations. I've been looking at wind observations from the buoy out in Mass Bay, so it's a little bit farther offshore, but it does still capture storms, which likely are hitting the bay as well. And I've seen really obvious strong trends in the increasing frequency of storms and the increasing duration and intensity of storms. And I'm just wondering if this could explain much of what's happening in terms of the eelgrass in those bays. And so I just wanted to float that idea just to kick off the conversation.
Mm-hmm.
Yeah, I'll jump in on that too. I think I had it on my slide, but I didn't say it out loud. That Catherine Ford and I saw that in our data analysis too. We were seeing from the weather buoys, we've seen significant increases in wind gusts, and also, interestingly, a directional change. So over time, I think it's becoming more northerly wind direction, which is interesting. So it could be pushing, sediment could be pushing around in a way it never has before. I really do think that stressor has some potential, and it could be a really valid one. It's hard to know what to tackle, how to get the data to back that up. And President and I have talked about maybe, deep coring to look for, sources of sediment. I'm not we have a sediment specialist on the line here. So,
Yeah.
we're all ears with all the other experts on the line.
Yeah, and actually, as part of our next steps, that's one of the things that I was going to talk about at the end that we will be doing. But we would be working with Brad, so we're bringing Brad into this conversation as well. So Brad, feel free to jump in on this.
Thank you. Yeah, so we are our plan is in August to take 2 sediment cores in locations that Jill and Cressida have identified as potentially shoaling in. And what our hope is to be able to use radioisotopes to get at what the sedimentation rates have been in that area, in those 2 areas. The other thing I'm thinking, listening to this, I'm wondering if there is any information in the past of what the sediment ground type has been. In other words, has muddy bottoms been increasing over time, or are they shifting? And then another angle that I'm kind of excited about that might shed some light on this is there are some radioisotopes like beryllium-7 that have such short half-lives that if you measure surface sediment and you have beryllium-7, it means that sediment has been deposited within the last year or so. So it could be kind of interesting for a future study to set up some sort of a grid pattern, or maybe target specific areas, take surface sediments and measure the beryllium-7, and that could give us an idea of influx of sediment versus erosional areas. And if some of these areas are actively shoaling in, we should be able to see that in some of those radioisotope data. So there's some thoughts that I have to share.
Phil, did you have a question?
Me?
Yeah, your hand is up, so I wasn't sure if you had a question.
I have lots of questions.
Good.
So I didn't. So I have a bunch of thoughts. So first, I think Forrest had rightfully mentioned water temperature as an issue, as a potential issue, and I'm wondering if anyone has data on that and what the mean water temperatures in the system are. So does anyone have that information?
Yeah, we do have that. There are a handful of a couple of Center for Coastal Studies stations measuring temperature within the embayment, and then there's a couple that are nearby, and DMF has some at depth stations as well. And between those and the nearest buoys, offshore buoys, there are patterns of significantly increasing surface temperatures.
Yeah, I don't care about the offshore buoys.
Yeah.
And I'm wondering, so could you just, and I'm not going to hold you to an absolute number or accuracy of quoting an absolute number, but do you know generally what the temperatures are? Like, are they significantly above 25 for extended periods of time?
No, not extended. The temperatures definitely creep in, and there are numerous days each summer that are spent above 25, but it's not, it doesn't seem to be prolonged. But it could be, I mean, who knows? How many days does eelgrass have to spend at 27 for it to be lethal in a well-fleshed embayment? I'm not sure. But yeah, when we looked at the data up to 2014, at least, there were definitely days spent in that danger zone.
Okay. So something you may want to consider for future, and maybe Jill or Sarah or someone who's been, or Forrest who have been out there can comment on this qualitatively. There's a theory that stressed plants will flower at a higher rate. So measuring flowering rates may help you discern whether this is a chronic stress issue or an acute issue that you're dealing with. So that may be something you want to think about. And that kind of feeds into the next, if I read one of your data slides correctly, the one on percent cover, and maybe I got this wrong, so feel free to correct me. In the 0% cover category, does that mean that 50% of the time after the year after you did have eelgrass?
Is this the right one first?
Yes.
Yeah, good question, Phil. The way that eelgrass cover was assessed at the stations, so at a single station, sorry, I'm going to go some background to help explain. So at a single station, there were 4 different drop camera points taken. And these were different assessments. And in patchy areas, those stations could have eelgrass present maybe at 1 or 2 of those drop camera points, but they are absent at those other drop camera points. So in terms of the station as a whole, that station would have been given a presence. Eelgrass is present at that station. We would have considered it present, even though in some of the drop camera points, it would have been, there would be no eelgrass observed. We get a 0% cover. So those are areas where basically the 0%, these are areas where eelgrass is present, but it's patchy. And then the next year, it's gone altogether.
Or it's filled in.
No, there's no gain in this data set. I've only been looking at losses or persistence. Yeah, so the orange is lost and the green is persistent. So I don't know, I guess, I don't know what the eelgrass cover was the next year in the persistent patches. This is just looking at the prior year. So there are some patches that are patchy. About half of the observations that are absent were in locations where the eelgrass did persist. So there is some locations where eelgrass persists where it's patchy, but less so than when it's more continuous or denser.
Okay. Then if you could go up to the leaf area index slide. So that's, I'm assuming that, so that's, you're taking individual shoots, so you're not accounting for shoot density with that on this slide.
That's correct. It's just the morphology of a single shoot. Yeah.
Right. And so if
I believe it's the largest leaf on that shoot as well. So it's the width at the stem and then the height of the longest leaf.
Right. And if you're in an area that's of low cover, the plants will not be self-shading, so they'll tend to grow larger. And so I really think if you want to use this indice, you should use it leaf area per meter squared, not on individual shoots, because that will give you a more population level response.
Yeah, I agree. I don't think we have the data to do it.
I know you don't, but I'm just saying if you're out there and have the ability to measure shoot density to integrate into this measurement, I think that would be useful. And then going to the epiphytes, what do you have a sense of the type of epiphytes? I mean, are we talking algae? Are we talking tunicates? Are we some mix? And was this taken in August, this measurement?
I can answer to the date it was taken, which the surveys were done in August. I believe the epiphytes are a mix of algae and tunicates. We do have photos. I have not looked through the photos in great detail to determine the composition.
Yeah, that's
There's
right,
some
Forrest.
hydroids too.
hydroids. And do you have a sense of like seasonal change in this component? I mean, obviously August is you know, a good time, but like out on the vineyard, we've measured this, and August, September, the plants are absolutely coated to the point where they're falling over. And yet somehow every year they come back. It just amazes me. And so I'm just wondering if you have a sense of like in the early fall, does it get a lot worse than this?
No, we don't know.
Because as
We can check.
I mean, the plants, obviously the way they stay ahead of epiphytes is leaf turnover rate, which is really a function of day length and sunlight. And as that decreases, you know, the epiphytes start to get the upper hand.
Yeah.
As water temperature increases, tunicates grow faster, et cetera, et cetera. So, so I don't know, you may want to think about just maybe adding another data point in the early fall on this. I agree with you though, Forrest. I don't think it's a major issue, but it would be interesting to just track that seasonally. And then on the Secchi depth, are those controlled for tide when those samples are taken? I mean, the range of data, well, one, with Secchi disc, you really need tons of data points to be able to say anything. And two, I'm just looking at the range in the persistent category, and it is fairly dramatic. And so again, I think that points to you really need a lot of data to be able to say anything quantitative, and you need to be able to control for tide and other parameters, and also the depth of your station. So the depth of your station obviously is
So, yeah. So the stations where the Secchi disc bottomed out were excluded from these analyses.
Yeah.
Because we couldn't tell what the depth was. It was some point beyond the bottom. And they were not controlled for tide. As Sarah mentioned, it's juggling the volunteers and when we can access the stations has been really meant that going out to visit every station at the same tide or consistent tide isn't feasible. I agree that there's many flaws to the Secchi data, and there's not a whole ton you can draw from this. Do you have ideas of another way to assess turbidity?
well, I don't know. You guys have unlimited number of hobos, don't you?
Fortunately not. I wish.
Yeah, I mean, I don't know, putting out a series of hobos, or I mean, even better would be power sensors, but
Yeah.
Those are cost prohibitive.
Yeah, Brett Habany, and sorry to bring you in at this point, but you did a lot of studies in Salem Harbor earlier on, around the same time, I think, but in 2012 or so, looking at turbidity. Is there something that could shed some insight onto what we can do? Maybe we can use the same methods here.
Yeah, absolutely. So we use turbidity sensors on multi-sensor probes. So we had some YSI probes, and then in the buoys we had a company called Manta, their probe, so similar to the YSI. So that's certainly something that we've done up, you know, in Salem Sound that we could certainly do. Turbidity sensors aren't that complicated. I think what we found in Salem Sound is if you're going out to do point measurements, you can calibrate really well that day, but trying to have the turbidity sensors, you know, on the buoys, they biofouled relatively quickly, even if they do have wipers.
Thanks.
Yeah. And Phil does bring up the, you know, the actual light sensors. He knows better than I do, you know, about the eelgrass, but I do understand that turbidity is not exactly the same as the light that, you know, the eelgrass are going to need. So I don't know as much about that as Phil, or I suppose Jill probably does.
And for the sediment stuff, has anyone looked at redox?
We're gonna
in
be
this-
doing that actually. So
Okay.
maybe before we I mean, as we discussion, maybe I should go, either we can continue with the discussion or I can tell you quickly what we have in mind for this year and next year in terms of monitoring. So why don't I do that super quick? So next steps, we have the Eelgrass Blitz coming up starting August 8th. Sarah is in charge of that and with Taylor, and it's going to be spread out a little more than usual. Usually we do it over a week, but because we figured it would be easier to get more volunteers and more boats if we spread it out over 2 or 3 weeks this summer. So stay tuned for that. So basically, we wanted to see going forward, you know, seeing all these results, etc. Is the KP still suitable for eelgrass, for example? You know, one of the questions. And we'll be launching a couple of surveys going forward. One is a water quality survey that we'll be working with EPA as to augment. So Center Coastal Studies has a lot of data, but there are a lot of their data around the shore, although in Plymouth, they also have offshore data as well. And we will be augmenting this to have some areas where volunteers go to, where our 119 or 120 points are. And we wanted to look more closely at these potential stressors, increasing temperature, especially nutrients and turbidity. And then also wanted to start delving into sediment sampling to assess physical and chemical condition and investigate any anecdotal accounts of burial. And then we also wanted to start looking again into an inventory of the potential sources of impact that kind of update the data that Jill and Catherine's report in 2016 had published. So in terms of the water quality, we'll be partnering with EPA Region 1. We'll be doing 5 stations this year. We're calling this year a pilot year, and we'll be sampling in August and September. We had planned to do more sampling, but other works and projects that EPA had already lined up precluded this. So we're hoping to be able to do more continuous proper sampling next year. But at the very least, this year we'll start kind of this pilot. We'll be measuring using a sonde for physical parameters and also collecting discrete data for nutrients and chlorophyll. Next year, we also be hoping to put in some hobos for temperature, maybe even DO to leave there over the summer. That way we'll be, you know, have a better idea on continuous data of the, you know, these changes across tides and across different weather situations, etc. And then the other part is the sediment characterization. We're partnering with Salem State University with Brett's lab here. We have 8 stations across the KP. There will be graphs from all of them, and 2 sites will also be collecting cores. We'll be looking for grain size analysis, also include measuring TOCs, TN, TSN isotopes. We'll be looking at redox and for the redox layer, and also that should be chronology, sorry. Using C0137 and radiocarbon dating. And that will be one sampling event in August. So there's going to be a lot happening in August. So just to give you an idea that we have these in mind, and that we will appreciate as we go through these questions, any feedback or input that you can have that will add to that. So sorry, I went a little bit backwards in the sequence, but I figured it's more useful.
Can I ask Brad a question? Brad, can you do bulk density in your sediment cores?
Absolutely, and that is standard what we normally do. So yeah, we didn't list that, but when we take our samples, we normally take a cubic centimeter and then get a mass both wet and dry on that. So yes, we can get that, Phil.
And for your organic carbon, how is that loss in ignition or elemental analysis?
Elemental, we have an elemental analyzer.
Yeah.
So we typically acidify the sample, you know, fumigate it to get rid of any carbonate that might be in there. And then you use our elemental analyzer to get the total carbon, or total organic carbon, I should say.
Yeah. So I would be really interested in that data.
Okay.
Especially, well, not so much in the unvegetated areas, but in vegetation. And we've done coring in multiple meadows in New England and have a regional database that we've accumulated now of that data. So I'd be interested in seeing that when it's available. Have to the sediment issue, has Brad or Sarah or anyone thought about use of sediment traps in the system?
It's a good question. I haven't thought about it. I have done sediment trap work before in Salem Sound, as well as many lakes I've done sediment trap work. From what I have found, it's a little bit hard in dynamic areas that have, decent currents moving through them. But it certainly is something that we could investigate. I don't know, Sarah, you may want to speak, and this is kind of your area. If you've done any of or thought about that at all.
No, I haven't. I haven't thought about that, but I'd certainly be happy to help.
Yeah.
Well, we've been using them in eelgrass in our studies, and I'd be willing to offer our equipment and assistance if that is of interest to this group.
I think that would be really great, Phil. Do you mind me asking briefly what kind of design you have for the sediment traps?
Yeah, so our Narragansett lab built them for us. They come in a series of 3 they're essentially PVC tubes, 5-to-1 ratio height to width, with a honeycomb insert to kind of break up the boundary layer.
Yep.
And we deploy them on the bottom, and we put in like tent stakes to hold them in place. And they work pretty well.
Yeah. So they're actually, they're anchored, staked to the bottom. That would alleviate a lot of the current issues that we've dealt with. So cool. All right. Thank you, Phil.
Thanks, Phil. Okay. I'm trying to see if there's any questions in the chat. Sorry.
There have been some chat comments. Maybe we can just make sure folks are reading those. One was about hobo light sensors from Dahlia, and the other is from Joanne about potential role of boating activity in causing sediment resuspension. Thank you.
So anybody can answer to that?
Todd has his hand up.
Yes, Todd.
All right, I wasn't going to comment on either of those 2 issues. I was just going to continue to talk about brainstorm on some other things. I like the idea about looking at temperature longer term. I actually just pulled up the Center for Coastal Studies data, and it's a little difficult to pull out from their data what might be happening exactly within the eelgrass bed. So yeah, if you could throw some hobos down there in both the vegetated and unvegetated, that might help get at that answer if it's warmer. And for how long? I like the idea that Jill had of, you touch 25C, but for how long? It might be helpful to kind of know that. And that might be, increasing if there's more shoaling. So it could be these 2 things are working together. You get more sediment, shallower water, it gets warmer, temperatures increasing.
Yeah, I know DMF had planned to put some hobos as well this year, but they might be wanting to move. I don't want to speak for you, Forrest, but I think you were planning also to possibly doing that next year as well.
Yeah, Todd, I had the same thought, that same kind of design. And we'd hope to maybe have time this year, but we didn't, and it was a low priority for us. So hopefully we can find some partners or some funding and maybe work on making that a reality next summer. Yeah.
Also, the other idea that Brad brought up about moving sediment and different types, like is there a mud patch moving around, or is there a sand patch moving around? That's fairly easy to do, sediment grabs and then sifting. And then I had one quick question for Brad. I see the other hands up. You talked about beryllium, but the proposal for this year or next year is to do cesium to look at chronology for time. Would it make sense to add the beryllium analysis to those cores?
It would potentially. I think the issue with beryllium in a sediment core is really what that's going to show you is if the very top of your core is active. So my thinking with the beryllium-7 would be more of a survey of the surface sediments to see which areas have active sedimentation and which areas don't. So we could certainly discuss, we're doing grab samples, in a handful of locations this year. So maybe we could do a little pilot this year and measure the beryllium in those, I think it's 9 samples. And
I think that makes sense.
Build on the following year if it looks okay.
Yeah, that sounds like a good idea. Thank you.
Yeah.
Yep. Barbara?
Yeah, I was just wondering about the sediment dynamics in the bays, and is there any way to compare, what is there charts from previous years, and can it be examined over the whole bay where these, where things are moving?
That's a good question. I know I have charts showing seafloor soft versus hard and different types, but I don't know if we have historic maps of that. Sarah or Jill, do you know how far that goes back?
I think it's a great idea to do that, to look to the old NOAA nautical charts, and then, to get decades old data. I don't know how easy it would be to analyze that if they're not sort of, workable GIS layers currently. They might just be static, but it's a starting point. And then more recently, there are some really good digital, I think they're LiDAR, bathymetry data sets for the area. So it wouldn't be too hard to look into those data sets.
USGS did more recently, as you said, Jill, did a lot of maps for CZM. And I believe the KP was covered as well. So what they did was they were stitching existing data sets that they thought were really good quality and kind of trying to fill in. So deep in the embayments, there were some gaps. So I want to take a look again at the maps to see if the KP, how much coverage that had.
Jill, I think Dan Sampson may have the GIS data for charts, because a few years back, he wanted to get all the asterisk rocks and he made a request. So I think when he got his request, I think he has all of the soundings.
Oh, cool. That's great.
I will check with Dan, thanks. I can't see everybody because my screen is up, but I think Phil and then Brad. So let me know if I'm missing somebody who's raising their hand, sorry.
So 2 things. One, on that last discussion, might we gain some good information from our good friends at the Corps of Engineers on sedimentation rates, at least in the navigation channels? I'm sure that they have some detailed measurements in those areas that may be worth looking at and getting that data. So that's one thing. The second is the number of hours over 25 degrees. From areas south of us in Peconic Estuary, it seems like there's a break point of about 1,000 hours per season. You get above that, and eelgrass abundance and presence is dramatically declined. I'm not sure that 1,000. It's not like the 10,000 hours of Malcolm Gladwell is makes you an expert, but I'm sure that there's some function of continuous number of hours in a row that something less than 1,000 that's probably critical as well. But we should keep that number in mind as kind of a guide. And my last point or question is, here you have listed eelgrass trench planting and latitudinal rescue. And so are we talking about something that is now referred to commonly as assisted migration, where we're taking plants from southern realms and moving them north in anticipation of increased water temperatures? So is that what you mean?
Yeah.
Yeah. So Brett, did you have a question? And then we'll move on to a little bit your thoughts about transplanting, the group's thoughts about transplanting, restoration, etc., where our investment endeavor should be. Brett, go first.
Yeah, the other angle I was just wondering, thinking about sedimentation and some of these areas that have mud coming in, I know that there are some USGS gauging stations on some of the inflows coming into this embayment. And I'm wondering if we might be able to start thinking about if either the discharge of these rivers have changed over time and or if land use has changed enough over time with development and such that we might be increasing the flux of fine sediments from the land into the embayment. I'm just playing very briefly on USGS site now, and I'm looking at the Eel River data, and it looks like it's discontinuous from 1969 to present. There's a big hiatus there. So I'm not seeing anything that's jumping out at me, but it might be an angle to investigate a little further.
Yeah, that's a really good point, Brad. And we did see significant increase in flow rate in the Center for Coastal Studies data. And I personally never connected that to mud being carried with the flow. That's a great point.
Yeah, yeah. Yeah, especially if you have clays coming down, fresh water, and then they hit that brackish water, they're going to flocculate and sediment out. So that might be part of the source.
Right. Okay, so in terms of one thing that DMF has been doing also over the past year or so is trying to see whether the KP might be a good candidate for eelgrass transplanting. So there are two things about that. One, losses might say, okay, transplanting is great. On the other hand, what are the losses attributed to? Does it mean that it would be a wasted effort or a better effort to, transplant or restore? It's grappling with that question as well, kind of. So we would like to hear your thoughts about that as well.
I feel possibly unqualified to comment on this, but it may be as simple as if you don't understand what the cause of the process is, it probably is premature to do transplanting to try to restore.
Yeah.
Yeah, let me just respond to that briefly, though, is that part of the problem is as eelgrass disappears from these areas, other uses have been moving in, especially aquaculture, mooring fields can move in. Once these areas are unvegetated, unfortunately, they're not really protected in perpetuity, even if they may still be potential habitat or suitable transplant sites. They end up getting displaced over time. So just to add that component in, we do see that. We have seen that especially in DKP with aquaculture moving in to areas that are all of a sudden devoid of eelgrass. So it's, as long as the aquaculture stays, it's most likely not going to revegetate there.
Interesting. Phil, you have your hand up.
I should just leave my hand up perpetually here. But I think it depends on what your goal for the transplanting is. I think there's value in, and I know Fred Short has said this in the past, so this is not an original idea on my part, but a lot of these restoration efforts start with pilot transplants. So you put out a test plot, and you can learn a lot from those test plots if you take the time to actually monitor them and collect the data. And so I think from that perspective, there's potentially great value in better understanding the system with test plots, which is the first step in restoration in many cases, than doing like a large scale, restoration effort. As long as you're willing to put the time in to collect the monitoring data post putting in the test plots. And then just a second point is now down in the Chesapeake, they don't do transplanting anymore. It's all seed-based. And we're starting to do more seed-based restoration up here, which is less labor intensive, and maybe something that we should be considering. I know Alyssa's been doing more out on Nantucket with seeds. Randall Hughes is doing more with seeds. My group's doing more with seeds. I think that should be considered in the mix.
Looks like Tay has her hand up.
Go ahead, Tay.
Hey, everybody. Sorry to not be fully in this meeting. I'm still really suffering from COVID. But I do want to chime in about the restoration and the test plots. I haven't given up on DKP. We did plant a test plot and it didn't really survive. And I have some ideas about why. I don't necessarily think that the site isn't good for restoration. I think we had some problems with the method in that case. And I agree with Phil. I think that doing small scale test plots with seeding is a good plan for DKP. I'm not giving up on that.
Thanks, Tay. Other comments, thoughts?
In terms of one other question we had is, and you guys, other presenters, please jump in, about the volunteer sampling or the volunteer project. It's something that we really enjoy doing every year. Is it something that we have been thinking of continuing it, of course, but do you guys have any thoughts on whether it could be continued annually or enhanced in some way, or, you know, any thoughts about that in general?
I think it's great that you accomplished as much as you have, and I think it's up to you as far as the level of interest you think you can sustain, and what it sounds like the limitation is the vessel availability, and it's up to you what puts constraints on it. But I not knowing what those constraints are, I might suggest that you think in terms of two aspects of the sampling. One, which would be routine, it happens every year. And then each year there could be one sort of special study or topic of interest where it's an opportunity to go get some non-routine sampling, and that's some particular topic that maybe you don't know anything about at all, and it would be great to just get some useful first data, because it's just the fleet going out there is just a great opportunity, I think.
Yeah. Yeah, and the other thing about it is that other groups and other, embayments have heard about it, that it's a growing interest. And I know Jill has been working with other places, both in, Cohasset, I think Salem Sound, Provincetown. So there's been, Jill, sorry, you can talk more about that. But yeah, we are excited about that because definitely it's a transferable method. And it's a lower cost way of keeping tabs more closely on eelgrass, with all its pros and cons, of course, but it's something that, especially because eelgrass sometimes is so ephemeral, and we cannot always afford doing acoustics every year and aerial service every year for all areas. So this is a way of, right, Jill? Yes. So yeah, so we'll be excited for people to be wanting to do this in their embankment.
So Dan, can I?
I was just inviting Barbara up, just
Okay,
if you have your hand up.
Yep, good. So yes, we are interested in doing it in Salem Sound. We started with a very small pilot, and we would like to continue doing that. I don't know anything how seeding is done, but I know that volunteers like to collect data, but they also like to feel like they're part of the solution. So if seeding is something that could be done at the same time while they're in the area, I don't know, but that might be an interesting way to involve people.
Yeah, definitely. We'll talk about that. So, okay, are there any thoughts or questions about this? Brad, I see you have your hand up.
Yeah, hi, thanks, President. I just have a quick question for my eelgrass colleagues that's hitting me here. Thinking about sediment grain size, the data that were presented today kind of binge things into, mud, sand, and sand plus mud. As a geologist, we normally do more in-depth grain size distributions to try to think about, is it, unimodal, bimodal, much more precise what the grain size is. I'm curious from an eelgrass point of view, is the binning that we saw today sufficient for information that you all need? Is it worth spending the extra time to use a laser diffraction grain size analyzer to get a much more in-depth, distribution of the sediment? I'm ignorant on that, so I'm curious if you could, I guess, educate me.
I think that's an important piece of the puzzle. In our publication on blue carbon, we found a definite correlation in grain size and amount of carbon in these systems. So I think having a finer cut of that would be useful. Now, I wouldn't necessarily put that at the top of the list. I would put water temperature at the top of my list, and then light data, and then sediment. So those three things, in my mind, are the controlling factors. And I don't know if it was Forrest or someone, or maybe it was Jill. I think you're not going to find a single smoking gun here. It's going to be a combination of factors. And it's going to be rising water temperature. It's going to be decreasing light and less friendly sediments. And the plants are getting to the point where they can't compensate for that cumulative amount of stress.
Right.
Thank you, Phil.
Thanks. Okay.
I just have a quick
We have some
question.
comments.
Oh, sorry.
Oh, sorry. I was just gonna say we have some comments in the chat there. Hope everybody's watching that. We see Tay is saying perhaps percent organic matter might be more important than grain size. Also, she mentions that at her restoration site, temperatures did not exceed 25 C, and they concluded it was not heat stressed.
Okay. Okay, any other thoughts or questions? I think you have given us a lot to think about. And it's like we were thinking along the same lines, but in terms of, best methods to use and how to address it, it's been really, really helpful. And we'll definitely be coming back to STAC for this as, more monitoring and stuff moves forward. So any thoughts that come to you after this and after break, please do share. We appreciate that. And yeah, and we'll let you know, as when we have some data, etc., we'll let you know how that goes, etc. We'll keep you in the loop on this as well. And then, do you think we need to should move to the next item? Or I don't want to cut the conversation short or anything like that.
Well, we're past 11:45. I think I'm getting a sense most people that wanted to, raise issues have been able to, which is great. And I think we can move on to the final part of the agenda. Unless folks have other things they'd like to bring up now.
Sarah, how do we volunteer for your effort?
That's great. I will, I'll put both links in the chat. There's a boat captain volunteer link, and then there's also a volunteer, regular passenger volunteer thing. So I will put those both in the chat.
Yeah, so you're definitely welcome to come over.
Yeah, come on down. It's look at my calendar. So it's August 8th through 12th, and then August 15th through 17th, and 22nd through 24th. And the time changes depending on the tides each day. So look at your calendar and the survey carefully.
Mhm.
So I'll post those right now.
Yes, great idea. Thank you, Sarah.
You take volunteers from the North Shore?
Yes,
Yes,
yes, we do. Yes, you do. Yes.
absolutely.
Okay, well, the rest of the agenda is